In previous classes, we learned about important biomicromolecules like DNA, polynucleotide, and RNA. Okay, the differences between DNA and RNA, and how these polynucleotides are made, and what are the linkages, and also about their structural properties. Okay, but how DNA and RNA are connected. Okay, so there are different types of RNA. So one of the RNA is messenger RNA. Okay. Okay. So messenger RNA is actually made from DNA. Okay. So using an enzyme called Rna polymerase. RNA polymerase actually uses DNA as a template and then makes RNA molecules. So because this is a messenger molecule, so to further code the protein, let's say it's called messenger, and it's messenger RNA because there are other types of RNA in the cell. So one is transferable RNA and another is ribosomal RNA. So to distinguish this RNA from that, we do that. So mRNA again translates information into protein on using enzyme ribosomes. Okay, so this is typical way in which the information is transferred inside the cell from which is stored in DNA into functional molecules or protein molecules. Okay, so again, one can actually convert one DNA molecule to another DNA molecule. You can convert sometimes RNA molecule back into DNA molecule, and you can make one RNA from another RNA. Okay, so there are a lot of actually different channels of information flow. So this is called central Dogma molecular biology. Okay, so how the information flow occurs from one polymolecule, biopolymer to another polymer is central dogma. Okay, so if you have to represent all this information in a simple diagram, so you write a DNA, so that's basically blueprint. So DNA can transfer information into another RNA. Okay, so that's using RNA polymerase. And then DNA itself can transfer into another DNA molecule. Basically, you make one copy from a template DNA, another copy. So similarly, you can actually make one RNA from another RNA. Okay, so for every process, you have different enzyme, and then RNA into protein. Okay, and rarely information goes from RNA to DNA. Okay, so you'll see where exactly it really occurs. So write DNA to DNA. Okay, it's basically DNA polymerase is the enzyme. Okay, our DNA polymerization is a process. DNA to RNA is DNA dependent RNA polymerase. So this is a process we call transcription. RNA can make another RNA. So here you have RNA dependent. RNA polymerase. RNA dependent RNA polymerase. You see, these two are two different processes, DNA to RNA with transcription, but RNA to RNA is RNA dependent RNA polymerase. So both are RNA polymerase, but they use different molecules or templates. In one case it's DNA, in one case it's RNA. Okay, and RNA to DNA. Okay, so this is called reverse transcription. Okay, because transcription is typically DNA to RNA. It's occurring opposite, so it's a reverse transcription, and you have reverse transcriptase as the enzyme, and then RNA to protein. Okay, so this we call transcription. Okay, so occurs on the ribosomes. So DNA to DNA typically in all cells, bacterial cells and eukaryotic cells. So one cell divides to another, so you need to make a copy of DNA. So DNA to RNA. So let's say we have to make proteins. Okay, so this is the intermediate step. So the template DNA is converted into mRNA transcription. RNA to RNA. Okay, so you make a copy of RNA. So when you make a copy of RNA, so typically this occurs in viruses. Okay, so in viruses, so these are RNA viruses where you have the genetic code of the virus is RNA. So then RNA invades the host, so then it needs to make a copy of itself. So then you have RNA polymerase, which actually make copies. Okay. Anyone have any example for virus which has this RNA virus which uses this mechanism?
COVID.
Hmm?
COVID.
Yeah, SARS-CoV-2, SARS-CoV, coronaviruses in general. Yes. Said something? Okay. Example: SARS-CoV-2. Okay, or basically coronavirus. So where the blueprint of the virus is just RNA strand. RNA strand is enveloped with lipids and protein molecule, and that invades a host. Then within the host, so basically that RNA can be directly used to make protein. Okay, so these are called positive strands. You don't have to really that's basically kind of quote unquote mRNA for the protein. At the same time, it makes a copy of themselves so that it can make more viral particles. So both ways of that. Okay, so RNA to DNA, reverse transcription. Okay, again, this is in viruses. Okay, so which viruses? So definitely it looks like it doesn't occur in SARS, because in SARS directly you make a copy of RNA using RNA polymerase, okay, to make a copy, and use ribosome to make protein. So definitely the information doesn't go back to RNA. But there are certain viruses where actually the genetic code of the virus is RNA. That RNA makes DNA, and then DNA is integrated into host genome so that it will be copied permanently. So whenever the cell divides now, that viral genome is actually transferred from one cell to another cell. Okay, unlike this virus where actually viral particles are made, but this is not integrated into cells. Okay, only the cells which have this this RNA will be replicated. But here, so reverse transcriptase. Okay. Example is HIV, human immunodeficiency virus. Okay, hepatitis B, retroviruses. These are called retroviruses. Okay, which use reverse transcription. Okay, so where RNA into DNA. Okay, so it integrates into host DNA. Okay, so example HIV. Okay, so there are many different types of viruses. For example, like that's are actually SARS-2 differs from HIV. Okay, in one case, you don't have this step in okay. So RNA to protein typically all cells, basically all living cells, either bacterial, eukaryotic, or prokaryotic, everywhere. So basically the proteins are actually made. So this entire thing is called central dogma. Okay, I'll show you the slide where actually this is depicted in a picture. Okay, everyone is seeing my slide, right?
Yes, sir.
Okay. Okay. So this is a central dogma. So where actually DNA can transfer information into RNA and RNA to protein. DNA can itself replicate in a polymerization reaction. RNA can also replicate. Rarely, again, RNA sends information back to DNA. Okay. So where it is marked blue and red. Okay. Blue is basically what really happens in our cells, normal way of living. And in special circumstances like viruses, where actually RNA can replicate itself, and RNA sometimes can make DNA molecule in, for example, like retroviruses. Okay, so this is a kind of mechanism first proposed by Francis Crick, 1957, almost immediately after the double-stranded DNA. So how exactly the information flow happening from DNA? Okay, how that DNA structure which they saw really actually helps this information. Okay, this is just a bit more picturesque. So where it shows that double-stranded DNA, and then polymerase DNA polymerase actually makes copy of DNA using DNA as the DNA. Just doesn't recognize my fingerprint because of the chalk dust. Okay. So RNA polymerase can transcribe information DNA to RNA. Okay, and ribosomes can translate this information to protein. Okay. So then, how the information from RNA to DNA to RNA is easy to understand because you have the DNA 4 nucleotides and then RNA corresponding nucleotides. Okay, but then how RNA to protein actually information is actually encoded. Okay, so there are a lot of efforts in, I would say, almost like 50s to 70s. Okay, 1950s to 70s.
I have a question.
Yes.
So, in case of RNA to RNA or RNA to DNA, these enzymes generally not present in our body
RNA to RNA.
or RNA to DNA.
For example, this is present in us. Okay, RNA to reverse transcription is present. Okay, in us, for example, I mentioned telomeres. Okay, so telomeres, if you read, basically as you age. Okay, so the chromosome length actually decreases. Okay. Second thing is that whenever cell division really occurs towards the end of the chromosome, so the filling is actually very important. Okay, to get the last nucleotide replicated. So the reverse transcriptases actually come very handy. So they actually add additional DNA fragments so that these are like capping agents, so that the protection, the chromosomes ends are really protected. So in that sense, when first proposed central dogma, the first slide. So the information was strictly that DNA to RNA and RNA to protein, but not back. Okay, so they never really thought that protein can really go back to RNA or RNA can go back to DNA. So strictly, that's not really true. Even like RNA to protein, and protein doesn't really code for RNA or DNA is not entirely correct, because in the end you need enzymes to even make copies. You might have template which serves as a copy, okay, but you need a machine which does it, like a Xerox machine. You need original transcript, okay, a document, but you need a Xerox machine to copy. Okay, so these protein molecules are like machines, Xerox machine, but then you need original copy to have this. Okay, so in that sense, for example, the reverse transcriptase is present in us, which actually codes back RNA to DNA. So these enzymes will actually present in us.
So when a virus enters, so does it just hacks this machinery?
Yes. So because most of the time it doesn't really have its machinery at all. Almost all the resources, except the blueprint, which it provides, almost all the resources come from the host cell. Okay, but rarely it codes for its a couple of important proteins only it codes. For example, like anything which is specific to viral genome. Okay, viral genome has a sequence. Let's say RNA. So it's not that entire RNA strand might code for protein. Sometimes it codes for the entire protein, but then it's not a single protein molecule. So these are fragments. So it needs to know when to stop. Okay, how many proteins to make. Okay, where to stop in ML. So that information doesn't really present in host cell because it's nothing to do with the host. So that information again passed through certain enzymes. So these are cleavage enzymes. I think in the case of SARS, I think we have S1 and S2, some enzymes there are, okay, which codes that, which actually cleave where exactly. So that information, that kind of information which is specific to viral genome is coded within the viral genome. So the corresponding proteins are actually made in the host cell. So okay, so in the 50s to 60s and even up to like 70s, so people are actually trying to understand how this RNA information, okay, encoded, finally translated into a protein sequence. Of course, we know that the ribosomes are involved, etc. But the molecular details were really absent. We know amino acid sequences, we know protein sequences, we know exactly how many amino acids are there, 20 amino acids. And we know what is a DNA consists of, 4 nucleotides, okay, and we know exact sequences. But how to actually match this information, okay, nucleotide to protein is not really. Everyone knows that, yes, you need corresponding DNA to make a certain protein. Then efforts from Corona, which he got Nobel Prize. Some of his efforts were try to make just polynucleotides, okay, poly A, poly C, okay, poly U, okay, poly G. or sometimes make doublets, okay, AC, 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 like that. You make variety of polynucleotides and then submit it to transferal RNA. Okay, so intermediate component I'll show you. So which is important to recognize a DNA sequence and then translate into corresponding amino acid. So it was just like a jigsaw puzzle. You make variety of sequences, okay, with some insight into how you are varying sequences, and then see what amino acids actually you are really getting. Okay, so they didn't really have to really synthesize the amino acids, but they know that something else, that there is a transfer of RNA. Okay, so for every amino acid, so this is the second type of RNA, tRNA. Okay, so typically in short form tRNA, and the amino acid, every tRNA codes for only one amino acid. So there is specific tRNA for every amino acid. For example, let's say alanine. Okay, amino acid, there is a tRNA alanine, or if it is a phenylalanine, then you have a tRNA phenylalanine. What you need to do is that what sequence of nucleotides actually bind this. That's all. If this binds to a certain nucleotide, okay, set, you know that nucleotide sequence would code for this. If a given nucleotide sequence doesn't bind this, means that there is a alanine coding nucleotide is not present in mRNA. So like that, you can actually submit a variety of sequences. You take, let's say, for alanine, you take different polynucleotides. Okay, it could be dimer, okay, oligomers. I mean, having two nucleotides, 3, 4, 5, etc. So people have literally done this in the 60s. Okay, finally putting all this information, which tRNA binds with sequence. So you finally find out, and what they found out, this code, this table, where this codon table shows. So this is mRNA, right? So mRNA binding to tRNA plus ribosome will give you protein sequence. Okay. So what is shown is that finally, of course, they found out that it's not one nucleotide. So you have, because only 4 varieties. So then you cannot have 20 amino acids coming from 4. So definitely it's minimum 2. Okay. So minimum 2 means again 4 into 4, only 16 varieties. So but you have 20 amino acids, and we know that every tRNA requires different sequence. So obviously you need to have at least minimum 3 nucleotides. Okay. So then it's 4 into 4 into 4. So total you have 16 into 4, 64 varieties. Okay. So then they thought, okay, let's begin with this. Let's not complicate. Okay. People always even thought maybe 4 or higher. But then let's say if you put 3 together. Okay. So you have 64 varieties of triplets. Okay. Then you submit all these 64 to every tRNA. You have 20 tRNA amino acids, and then see which one actually binds which. Automatically you have this table. Okay. So for example, let's say the top left, if you see UUU, so coming from which DNA sequence? So this is mRNA. Okay. So it needs to come from DNA. So what is the DNA sequence which give you UUU mRNA or RNA? AAA.
A.
Okay. Are you sure?
Yes, because in RNA it's U, but in DNA it's T, complementary to A, so it must be AA.
Okay. So yes. So you have AAA, so which is given for mRNA UUU. Okay. So let's say UUU is basically a triplet mRNA. So which shows that here it codes for phenylalanine. Means that if you have a transfer RNA which codes for phenylalanine binds to this. Okay. but we have only 20 amino acids. Okay, so some transfer RNAs. Okay, there is a what do you call redundancy, right? Okay, so you have actually tRNAs which actually bind two sequences. I mean, right? There are two different phenylalanines. Okay, so means that you have tRNA phenylalanines. So you have actually two types of transferable RNA for phenylalanine. So one binds UUU, another binds UUC. Okay, so this is redundancy. Means that somehow phenylalanine coding tRNAs, there are two varieties. Okay, so one binds UUU, another binds UUC. Okay, so like that, if you let's say go for alanine, okay, so then it is GCU and GCC. Okay, so there are GCU, there are actually 4 codons which are actually coding for alanine. Means that there are 4 tRNAs, okay, so which recognize these four different. Means that somehow you have variety. Some amino acids have more codons, some have less. For example, let's say which one have only one. Okay, so methionine AUG. Okay, AUG is there only one codon. Okay, and the highest is 4. So alanine 4, threonine 4, valine 4. Okay, leucine there are 6. Amazing, right? Proline there are 4, serine there are 4, histidine 2, glutamine 2, etc. Okay, glycine 4. So arginine again there are 6. Okay, shows that some amino acids, okay, more codons, means that somehow this looks like kind of through evolution. So let's say one amino acid is incorporated higher than other. Okay, so then somehow they are variety. Okay, so it looks like this is only evolved through. It's not that there are many known rules that why one has more than the other. Looks like some amino acids want more variety. And if you see, interestingly, 3 codons, UAA, UAG, and UGA. The 3 codons do not really code for any protein. Okay, do not code for any amino acid. But however, the important thing is that they serve as a stop. Okay, so you have a long mRNA strand, okay, which is derived from your DNA, and somehow it goes beyond what is required for protein synthesis or protein sequence. So these codons actually help to tell the ribosome that protein sequence is actually ended, so you stop synthesis. Beyond this point, you don't really need to synthesize. So again, you have tRNAs. Okay, so tRNAs for stop as well. Okay, so this again tRNAs which detect. Okay, so this codon and do not have any amino acid data set. Okay, so they don't have any amino acid. So so that nothing to add to the growing polypeptide chain. So basically protein synthesis stops. Okay. So, but of course,
sir, I have a question. That
yes.
These codons are attaching to the tRNA to identify the amino acid sequence, to detect the amino acid sequence. So how these are attaching with the tRNA? Is there some specific base pairing or other sort of chemical reactions?
No. So tRNA, okay, so tRNA now it's not just purely RNA. The moment I said that it has an amino acid attached. So this is basically a bioconjugated molecule, you can say, an RNA molecule, somehow at one end, okay, alanine amino acid is attached. It's neither purely amino acid, not purely RNA. So it's a conjugated molecule. So which itself is made by a mechanism where you need RNA molecule, you need an amino acid, where exactly it is attached. And also this RNA sequence might be different from one tRNA to another. So that's made entirely separately. So these are again small building blocks made parallelly inside in this. Okay. So interestingly, so this is originally this is the codon table which is used both in prokaryotes and eukaryotes to make protein molecules using ribosomes and mRNA template and tRNAs. Okay, so now we know so much of this. We actually artificially change this at will to incorporate, okay, artificial amino acids, artificial or unnatural amino acids into sequences. Okay, what you all need is that, so you need a tRNA, okay, so which recognizes a codon, but at will you change that amino acid to, for example, let's say another, let's say some function group added, okay, modified amino acid. So if you can make a tRNA using the entire sequence of tRNA and RNA, but however just change this amino acid with a certain functional group, or it could be modified amino acid, because then the recognition element is coming from RNA. Okay, so once that RNA recognizes the template, it binds to ribosome, and then as long as this is an amino acid, alpha amino acid, that can nicely transfer to the growing molecule fraction. So actually, people have been really doing this artificial amino acids incorporation into protein sequences, so that we can really see the changes. For example, a small modification on a big molecule. How does it affect the structure of the biomolecule, protein, and how does it affect even the function, binding, you name it, either in vitro context or in vivo context. You can actually do these tricks inside the cell. So there are cell lines what are called octopus. Okay. So, okay. So for example, where you can deplete certain tRNAs, okay, which are present in natural systems, and then you can actually add the one, okay, changing. Okay, so you can actually insert the tRNAs which you need. Okay, so of course modified amino acids. So then you can actually insert the amino acids. So people have been really doing that. Okay, so let's say how to this understand a little bit more. Okay, this table is like a periodic. What is a periodic table to chemistry is basically this codon table to geneticists. Okay, so for example, this sequence. Okay, you have an amino acid sequence: glycine, leucine, proline, tryptophan, glutamic acid, aspartic acid, methionine, tryptophan, phenylalanine, valine, and arginine. Okay, so from this table now you can actually write. Okay, so what are the possible templates which might have been used to get this protein sequence? Okay, so for example, like leucine, we have 6 codons, right? So all are actually added. So for every amino acid below, the possible codons are added. Okay. So now let's see if I have to ask you that what is the mRNA template which has been used to synthesize this polypeptide. You can have many different combinations. Okay, because for example, like leucine, you can use any of those 6. For glutamic acid, you can use any of those 2. For tryptophan, of course, only 1. For methionine, only 1. Okay. So in the intermediate region where it says the region of minimal degeneracy. Okay. So just for this sequence, tryptophan to valine. Okay. So how many possibilities? Okay. So glutamic acid, we have 2 possibilities, and aspartic acid, we have 2 possibilities. Phenylalanine 2. Okay. So so then you have basically. There are 8 possible, okay? So 8 possible sequences to code that peptide. Okay? So if I have to ask you, right, what is a mRNA possible template? You can come up with 8 possible templates. Any of those 8 would give you exactly the same, okay, sequence. So this is called sometimes redundancy because you have only 20 amino acids and you have 64 codons. Okay. So because of this redundancy, is again you can really play with this. Okay. So like for example, like phenylalanine has 2 tRNAs. So selectively you can actually knock out 1 tRNA, and you can use that codon for your artificial amino acid. Okay. So these platforms are actually now a reality. People have been able to. Okay, so that's delving into this. Okay, so yeah.
How will they ensure that it doesn't be three bases that hold for one amino acid, 0.4, 5, 6, 7?
No. So that's basically yeah. So initially it didn't really know. Okay, but now you have to really see that. Let's say if it is not 3, 4, how do you rule out? Okay, so just using poly-U is not going to help you because poly-U will give you always phenylalanine, okay, polyphenol. But then let's say if you couple, let's say if you use 12 codons, okay. So imagine, let's say these, okay, these 4, okay. So if it is 4 codons are used for an amino acid, at most you should have a tripeptide. Okay, but somehow you ended up having 4 amino acids. Means already it shows that, okay, so you already have 3 is enough. Okay, so these kind of tricks they have really found. Okay, so whether it is 2 to 4. So if you read Corona's work, he has actually used dinucleotides making poly like AC, okay, or poly-U, okay, poly-A, variety, okay. So he made many different. That's actually his contribution. How to make synthetically oligonucleotides of a known sequence, so that you can use them with tRNA binding, so that you know that because tRNAs could be really separated, okay. So that really actually led to that you need 3 codons are actually good enough. But artificially, okay, people have been actually doing this. Okay, so if you use 4, okay, so then of course you will have much more variety, right? So 64 into 4, okay, you can really think of 256, I guess. Okay, yeah, okay. So, but the thing is that it's only for artificial things. But natural things looks like 20 amino acids is good. Yeah. Yes.
So it's about reverse transcriptase. So we know that RNA is less stable than DNA from central dogma, actually. So RNA is less stable than DNA. Then what are the possible reasons of virus having RNA as the genetic material and then again reverse transcripting it to DNA, then forming RNA? It's a long process after all. So why can't, means what are the possible reasons? I know that there's the basis of evolution into it, but still, what are the possible reasons of RNA being the genetic material for some viruses? I mean, retroviruses.
Yeah, I don't know exact answer because I'm not really a virologist. But looks like, I mean, see, the microbes, okay, are means germs, okay, generally. But viral particles are only specific class of that, okay. So looks like they need to have, I think, minimum amount of information. Okay, possibly maybe directly usable by maybe, I don't know, post missionary, you know, as soon as they enter, okay, to use that maybe directly to copy information right away, okay. But the reverse transcriptase somehow it doesn't really work because somehow it's like kind of little capital investment for them. They say that, okay, it's not that right away use my RNA to make protein. But however, it takes a little longer time, but insert into host genome, okay, so that the gains are actually large. So once it is inserted in one cell genome, whenever that cell actually divides, the viral genome is actually divided, okay. And once it is again in the genome, you know, so depending on the transcription occurs, you know, so that machinery can be replicated. So I don't know why some viruses, for example, reverse transcriptases, actually went through this stuff. Looks like they are also trying different ways of actually maximizing their effect, okay, on hosts. I think that's the short answer I can give you, but I'm not really sure. Okay, someone said that in biology, evolution is the only way to explain why certain things actually do really happen. So it looks like many other different varieties of processes are actually mechanisms are worked out, whichever actually is beneficial is taken for. Okay, sorry, Suman, that's the answer I can give you.
Okay, thank you, sir. That's fine.
So now we know a little bit more about how the information from DNA to RNA occurs. That simple structure really helps. But RNA to protein is through interesting triplet codon cable. Okay. So, but it's more than this, So it's not that always. So DNA to RNA, okay. So every base pair is converted into every base is converted into another nucleotide in RNA. Okay, it's basically one-to-one. Okay, so let's say if you have 5 bases. Okay, so then you have pentanucleotide. Okay, so that's how actually DNA polymerase really work. Okay, so but when it comes to proteins, though we know that it's triplet codons, So let's say mRNA has, let's say, 300, okay, nucleotides long. So then typically, based on the codon table, what is the polypeptide chain length?
100 it will be.
Okay, so polypeptide chain length is 100 amino acids, right? So that's what you would expect, because that's what his codon table says, that every 3 nucleotide you have amino acid. Okay, but this is not always true. Okay, so this is not always true for the reasons is that mRNA has sequence. If you have to write, this is a sequence. Okay, so 5' to 3'. So there are actually elements, So some elements are actually not coding for protein. Okay, so this codes for protein sequence, this codes for protein sequence, this codes, but this intermediate segments, this doesn't code for a protein. This doesn't code for a protein. This doesn't code for a protein. Okay, so the elements or the sequence stretches which code for proteins are called exons. Okay. Okay. Again, all these 3 are to trial and error. You know that mRNA sequence is so long, but you are not getting protein so long. But then if you see carefully codons, you can actually match the amino acids to stretches. So the elements which don't code for protein are called introns. Okay, so mRNA, now you can really think of pieces of exons and introns. So exons are the protein coding regions and introns are non-coding. Means that DNA has a sequence where certain stretches actually don't really code in the end for protein. Okay.
Sir, I have a question.
Yes.
Sir, is the reason of not generating a protein, is it due to the surrounding factor, or is it that intrinsically certain triplets are there which doesn't have any protein? My question is because when you showed the codons list, we had 64 triplets.
Correct.
So 64 is the maximum possible triplets that we can obtain using 3 nucleotides,
Yes.
right?
Yes.
So it all of them has a protein to express, no? All of them have an amino acid to express.
Ideally, yes, they do.
Yeah. So why these regions?
Exactly. So that's what actually I explained. Okay.
Okay.
So, okay. So we know that these pieces which code for protein are exons. Okay. So then, so this is called pre-mRNA. Okay. Okay, pre-mRNA. And somehow it shows that now you cannot really submit this mRNA directly to ribosome. So before actually submitting to ribosome, there is some sort of pre-processing. And if someone knows programming, they'll understand what is pre-processing. Okay, there is something like pre-processor. Okay, so what happens in pre-processing? You actually remove introns. Remove introns and stitch together. Stitch exons. Okay, so this is an important step, typically, which is not really written in simple central dogma. Okay, so in this pre-processing, you remove introns and stitch back these exons. Okay. Okay, what is the advantage? Why this additional pre-processing step is required? By the way, so this is only present in eukaryotes. Prokaryotes, you don't have introns. Prokaryotes, you have entire mRNA, which comes out of your DNA, it directly goes into ribosomes. So only eukaryotes have additional RNA. So whenever eukaryotes means make things complex, means that there is an underlying, okay, underlying principle that somehow it enriches something else at the expense of spending energy, right? Okay, so what is the advantage? Okay, so interestingly, when you stitch exons together, you can have, interestingly, you can have actually variety. Okay, you can have, let's say, let me write exon as just ex1 plus ex2 plus ex3 plus ex4. That's one way I can stitch. I can stitch ex1 plus ex3 plus ex4. I can have ex2 plus ex3 plus ex4, so on and so forth. Okay, that's what actually the stitching really happens. So now you have just from one gene, you have variety of protein sequences. Okay, so you have just from one RNA, you can have a protein having all exons, or protein having slightly less number of exons, means that your polypeptide sequence is different. Okay, something is missing. Okay, maybe this is a totally different protein. Okay, basically any property which comes from exon 2 is not there, but that's a totally different protein. Okay, so you can have now many different proteins. Okay, from single gene. That's the interesting of introns and exons. Okay, I think this slide depicts that. Okay, this process is called splicing. Okay, this is pre-processing or actually splicing.
But why do you need to intron first? Why do you need to synthesize introns? So let's say if you want to go for like this different varieties of proteins, you can cell can directly synthesize the mRNA exon 1 plus exon 2 plus exon 3 plus exon 2. And then you just remove whichever you want.
No, but then you need to see, interestingly, when which parts of DNA are made mRNA have in starting elements. Now you need to have that starting element for every exon. Okay, that's redundant then. But here, using just one place, okay, so later step, okay, so there are again enzymes which participate in detecting introns and then doing this job. That itself is complex. Okay, looks like so it's advantageous to have rather than having exon 1, 2, 3, 4 as totally different proteins. Okay, so then they are expressed as different. So then you have to stitch proteins together.
No, my point was not that. My
Okay.
point was that from mRNA, you ensure from DNA, only exon 1 plus exon 2 plus exon 3 plus exon 4 is synthesized.
On ribosome.
No, from DNA from DNA to mRNA.
No, but you cannot. Okay, so which enzyme uses this? DNA-dependent RNA polymerase.
Yeah.
So you're telling RNA polymerase, no, don't synthesize this. Jump onto this.
Yeah,
Okay, to exon 2. But that doesn't make it. That's not how it happens. So it cannot really jump because it just faithfully amplifies the DNA as a template. So it doesn't know, it doesn't even care whether it's exon or intron.
This essentially means that whatever part of DNA goes for the intron sections, those are just a waste part.
Waste part, but the thing is that that's actually paying off later. Okay, so you have these additional elements. So again, introns, if you look carefully, there are sequences which are recognized by what's called splicing. Okay, so this process is called splicing, that removing the introns and stitching exons, and the enzymes are the complexes which are splicing. Okay, so these enzyme molecules detect from the intron sequences. Obviously, some sequence information is going. I don't think it's exactly known how it's read. Okay, so that sequences are actually important here. Okay, so it looks like maybe this is a better way than like bacterial way of making exons as independent protein. Then of course concentration is important. How they are stitched together important. Okay, and second important thing is that. So you see the sequence of 1, 2, 3, 4. So there are always like 1, 2, 3, 4, 1, 2, 4, 1, 3, 4, 2, 3, 4, 1. Okay, so like this. Okay, but it's never 4, 3, 1, never. Okay, so 3, 2, 4. So you see that order is actually maintained. So the exons order is maintained, though you eliminate some in between. Okay, that's an important. So how actually they stitch together. So this is important. So they always have this sequence, okay, which are present in this. But never you don't reverse the occurrence of exons. Okay, so this picture shows.
So, sir, excuse me, sir.
Yes.
So, sir, how can we differentiate introns and that stop codons?
No, stop codon is, see, no. So this introns and exons business, you do not have ribosome yet. So ribosome comes only after you stitch your exons, right? So ribosome, by the time the sequence comes to ribosome, every nucleotide codes for amino acid. Every nucleotide in triplet is involved. Okay, so introns are not there by the time it comes to ribosome. So that question doesn't arise because ribosome, whatever is submitted to the ribosome, is synthesized perfect.
Sir, I have a question.
Yeah.
Sir, earlier I asked you that what is the differentiating factor in, let's say, heart cell or lung cell? So, is this the reason that despite having the same DNA sequence, it expresses certain genes? You told that both the kind of cell expresses differently. So,
Yeah.
is this the differentiating mechanism?
Yeah, so this is partly true. For example, like certain molecules, change as you age. Okay, during initial development versus the later growth, the molecular composition change. Okay, so this intron and exons, so typically are important for very large protein molecules. Okay, so very often problem is that in laboratories we study very small protein molecules, but most of the proteins are actually come in what are called tandem manner. They are attached to end to end, and also this exon varieties. Okay, so especially for example larger proteins, especially proteins in the muscle. Okay, so they are so large, megadaltons of size. So they have exons of even 30, 40 exons are together. Okay. So how this large protein molecule, whether certain exons are present or not, gives some beneficial strength during the early development versus later age, for example, organisms or adults. So this is partly true. Your question that this exon business actually come into effect to some extent. Okay. So coming back to splicing. So
Sir, I have a question that you said that in viruses they do not have that introns part. In eukaryotes we have the only that introns parts and exon parts. So eukaryotes, the body is much more complex than the prokaryotes.
Yeah.
And so we need much more proteins molecules to regulate our biological process. Is it the reason to the process splicing came into the account in our system, not in the prokaryotes?
No, I mean it's difficult to answer because if you see the mustard plant, Arabidopsis, scientific name, Thaliana, actually has more genes than you and me. Surprisingly, a lot more genes than you and me. And somehow our genome size I think is slightly larger, if I remember, or maybe its genome is larger. But definitely, the genome means that are the genes, number of genes present in plants are higher than us. Okay.
Okay, sir.
So again, saying that this is one of the ways maybe that's helpful. You know. So let's say even if you have one gene, okay, but the thing is that one gene codes for 20 different proteins. Okay.
Okay.
So that's how actually you change the protein specificity or protein content or protein structure. Okay. So looks like exons might be important. Okay. So let me finish. So this process is called splicing. This is an important step, but often ignored because always people think that mRNA is directly coded to protein in the central dogma. Often people miss that there is intermediate step. Okay. At the top picture shows that a pol2, it's a polymerase, RNA polymerase, C binding to a DNA strand and synthesizing RNA. Okay. So RNA is synthesized from 5' to 3'. You have green exon and introns in it. So in the second step, okay, all these green regions are actually exons. And then this, can I call this yellow regions? So these yellow regions are actually non-coding. So in this step, okay, so you have spliceosome coming into picture and remove these introns and then stitching these DNA together to give you a template which can directly be now given to ribosome to synthesize protein. Of course, I'm not really going to too technical thing because there are some things like you need to add polyadenine at the end and so on and so forth, so that it doesn't really code for protein, but it's basically it will provide as a handle, okay, for the ribosome, etc. Okay, just before we stop. Okay, so this is another slide showing how exactly that might be useful. Okay, so this what I said is alternative splicing. Where you stitch together exons coming from the same gene, okay, in a different order or different number of exons. Beautifully color coded here. It shows that 3 protein molecules A and B, which are different from each other because they have different amino acid sequence in the end, coming from the same DNA. Okay, so DNA copied into RNA. Okay, so you can call that a pre-RNA or pre-mRNA, which is faithfully copied from the DNA. Okay, using what is the enzyme?
Speciosome.
No,
DNA dependent RNA polymerase.
DNA
Polymerase.
RNA polymerase.
DNA dependent RNA polymerase.
because there is RNA dependent RNA. Okay, so then you have exactly the same size as DNA. Now, this splicing splicing or alternative splicing will give you 5 exons. Okay, in protein A, you have all 5 exons stitched together 1, 2, 3, 4, 5. But protein B, the exon 3 is missing. You have 1, 2, or 4, 5 in that order. And so between protein A and protein B, the protein sequence coming from this exon 3 is actually missing. And in protein C, exon 4 is missing. Okay, so it's a different protein sequence. So this is actually picture shows that. So when we call mRNA, so we automatically assume that spliced mRNA in case of eukaryotes. But if it is prokaryotes, you don't have to mention because there are no introns in prokaryotes. Okay, you have to.
Sir, I have a quick question. Sir.
Yeah, go ahead.
Yes, sir. So for the formation of these 3 proteins, A, B, and C, the same DNA sequence has to be transcribed at least 3 times, no? Here. Because we need 3 mRNA for making 3 possible combinations.
No. So imagine, let's say one polymerase is making, let's say, 10 copies of RNA from DNA.
Okay.
Okay. So these 10 copies now you divide 3 into 3 into 2. Let's say first 3 have spliced to get A, another 3 spliced to get B, and then remaining 4 spliced to get C. Okay.
Okay, sir. Yes, sir.
And maybe this is another way to address your question that why the advantages. Okay, so you have one mRNA, but you already have 3 different products, okay. But in a controlled manner. So if we don't have this control and the proteins need to be stitched together, then it's concentration dependent, space, and whatnot. A lot of other things. Okay, so I think I gave you some overview about splicing because that's I thought chemistry students should know because this is an important process which is not really taught. Okay, so more detail. Okay, now we know what is mRNA. So when we call now mRNA, so now we know that it's basically spliced or RNA spliced. Now this mRNA is submitted to ribosome. Okay, now the codon table is active. Okay, now the codon table, you need to look at the codon table. The ribosome as actually is a multimeric complex consisting of RNA called ribosomal RNA and plus lot of other proteins. Okay, the assembly itself has many other factors to assemble one ribosome. Okay, so typically ribosomes come as so now. Okay, this rRNA is to distinguish from mRNA and tRNA. Okay. So typically ribosomes, if you see both eukaryotes and prokaryotes, so typically they come in kind of 2 fragments. Okay, and so typically one large subunit and one small subunit. So if you take the bacterial one, it's basically 50S and 30S. Okay, S is for Swerdberg unit, which you need to read what it means. Basically, it's not the molecular weight. Okay, it's basically when you centrifuge these molecules, they come at the different levels in a known viscosity solvent. So where actually they will come, will give you that number. Okay. So 2 subunits, okay, so come together, assemble on an mRNA. Okay, so mRNA has single strand on which these subunits assemble, and then they start reading now codons, 3 nucleotides at a time. So once 3 nucleotides are there, so the detector region where they will bring in, depending on what the codon, they bring on the corresponding tRNA. Okay. So tRNA, okay, let's say alanine. Okay. So the corresponding transferal RNA will match this codon, will come and bind. Okay, so this is where the matching is very, very important. Okay, now imagine so how accurate the ribosome needs to be. Okay, so each ribosome making many protein molecules at a time. Okay, so if this process is not efficient, if the detection efficiency is not close to 100%, you will have proteins which are not correct in sequence. If they are not correct in sequence, of course, we know what really happens. Okay, anything which you can't explain, aggregation, misfolding, you name it, non-functional unit, the cell filling with unused material, everything. Okay, so ribosomes need to be really accurate in detecting that codon and bringing in the right tRNA so that you have right amino acid. Okay, so of course there should be a start. So there is a start codon. Typically the synthesis occurred at methionine, the codon in AUG. So that's why if you work in protein labs, typically they always put AUG in the first codon for the any protein sequence because that's the starting. Of course, some other elements will be there. Some other elements will be there before 5 prime, which are important, etc. I'm not really getting into. Yes, there was a question.
Yeah, so the tRNA comes on the 50s or the 30s because in the slide it shows that it's coming in the 50s subunit.
So okay, so don't worry about those things, you know. So don't worry about those nitty-gritties. Okay, so basically both. It's not again that tRNA interacts only with one subunit. No. This is a 3-dimensional picture. It's changed into 2 dimensions, so it's super simplified. Okay, so but of course both units are actually important, both subunits. But tRNA comes and then detects this triplet. Only this tRNA right one will come and detect, and then the lot of ATP dependent motions, lot of ATP energies consumed. Okay. And a lot of macromolecular dynamics that happens on the this ribosome with mRNA. So mRNA needs to slide, push, pull, you name it, and then amino acid is attached. Then amino acid has an exit channel. Okay. So, but in the end, so once the right tRNA comes, that amino acid is actually incorporated. The first one is nothing to attach anything, but the second one comes. So then the second triplet, this actually slides. Okay, so that's mRNA movement in the machine, or you can think of ribosome moving along mRNA. Okay, so when it comes to the next triplet, so second tRNA comes, and then second tRNA comes with another amino acid, let's say phenylalanine. Then phenylalanine actually now covalently links with alanine, making the peptide. Okay, so basically you need to eliminate water. You have NH3 and COOH. So you eliminate the water and you make the peptide bond. And then now you push this dipeptide to bring in third codon, third element. On the right side, you see the sites, E site, P site, A site. All these, a lot of work has been really done, especially using single molecule fluorescence biology to understand these dynamical motions in the ribosome, how exactly the sliding of mRNA occurs, how the sliding of polypeptide really occurs, okay, and finally the peptide is made. Okay. So once the last codon is stop codon, then the corresponding tRNA doesn't really bring any amino acid, no peptide bond to make, and then the polypeptide just comes out of the exit channel. Okay. So, and also what you need to really imagine is that amplification is important. You have one DNA copy, and then you make a couple of mRNA copies. But just even if you have one mRNA copy, so let's say mRNA is long strands. Okay. So ribosomes come and okay, at different sites. Okay, not different sites, I mean, so once the first ribosome moves, okay, so 5' end comes, second ribosome binds, and then second moves, you can have actually third and so on and so forth. You have actually many ribosomes working on a strand. Okay, so here actually a lot of polypeptide chains, here shorter, here even shorter, here shorter. So as the ribosomes move along the mRNA strand, you can imagine that the ones towards the end already larger chain coming up, the one at the beginning just okay. These are sometimes called polysomes. Just for the name, that there are many ribosomes bound to RNA, and these structures, ribosomes are so big, you can actually these structures directly in electron microscope, even primitive electron microscopes. Okay, in 60s and 70s, directly could really see that mRNA actually coated with nicely ribosomes called polysomes shows that again ribosomes move from 5' to 3'. That's basically basically converges to now the important world of protein molecules. So now we have learned basically the cell components, important structures, organelles inside the cell, important macromolecules, DNA and RNA. We have learned structure of DNA and RNA, and then we learned how the information is transfers from dRNA, what are the molecules are involved in translating this information from DNA to RNA and RNA to protein. So now, so we will begin basically the world of protein molecules, which is much more complex than DNA and RNA, and that's because all the work which is done inside the cell, almost all the work. Okay, so some are the other protein element is involved. Okay, either it converts directly the chemical energy produced because of the reaction, using that energy to drive something else, or it basically uses ATP energy. Okay, so to drive, for example, machines like ribosomes. So in almost all aspects of living organisms, proteins are important nanomolecular machines that drive at the end of the day the work. And so it's important to understand what do they consist of. Okay, so what the proteins do really consist of, what are their sequences, why there is a variety, and for a given sequence, do we know what structure it will take and why it will take and how it will take, how the structure might be important for the function of the molecule. Okay, so in both sense. So I think we delve into that. So then again, we need to start with the basics. Okay, so amino acids. So proteins are nothing but a polypeptidic bond or polymers of yeah. Okay.
So it's the same RNA sequence. Can it act as mRNA, tRNA, or RNA?
No, because the tRNA, if you see, so tRNAs have actually very specific structure. Okay, so somehow often they will show something like this. Okay, so here more or less all of them kind of this shape. So to some extent, secondary structure is no. mRNA is very complicated. Okay, mRNA can't really predict because that depends on basically different proteins. You have different sequences. And what is ribosomal RNA is fixed. Basically, what it consists of. Okay, but only mRNA it's difficult to predict. Okay, but tRNA more or less you have 20 tRNAs, maybe more actually. We don't know. But structure is more or like this, and ribosomal RNA is ribosome. So ribosome structure is this. But ribosome is much more complex. We have ribosomal RNA molecule and protein molecule. They just not self-assemble. You need actually adapter molecules. Okay, there are hundreds of adapter molecules which make them these machines. But once they are assembled, they are stable. Okay, at any point of time there are thousands of ribosomes. I think per minute or something assembled inside the cell. Okay, there are some numbers.
And all the 20 RNAs, they also have the structures different from each other.
Sequences will be different because that's how actually you need to detect. Okay, because this tRNA, okay, somehow detecting the codon is opposite. Okay, in addition to that, something else is also in the ribosome. Maybe towards this, maybe similarities, but this and definitely they are different a lot from each other. But all will look kind of T-shaped.
Means something should be there in the structure itself, which will dictate that you have to bind to this anticodon and then to this amino acid.
Yeah.
If all are same, then how will they bind to this?
No. So this here, definitely this is complementary to this. First thing, absolutely complementary. But in the neighborhood, the sequences will be different. Okay, but they can't be so different that this will not really bind. Finally, this needs to recognize not only this codon, but it needs to recognize ribosome as well. Okay, the differences will be there definitely at this position. But I presume this region it just provides this structure. You know. So finally, what's important is that how the amino acid is placed. Okay. So this you have two tRNAs. Okay. So a growing chain of mRNA. So the tRNA binding is important, but then placing the amino acid proximity is very, very important because you need to bring, let's say this is growing chain, right? So this is a carboxyl of this amino acid needs to be close to amino acid. So that the reaction needs to facilitate so that you remove your molecule peptide bond is formed. And then everything else is actually again moving. Okay. So again, you need some globally structural similarity because finally all need to come out from the ribosome. Okay. Again, ribosome cannot really accommodate grossly different shapes. So the overall shape might be similar, but the recognition element is definitely this is very, very important. And then neighborhoods may be similar. But the question you're asking is that if I have to overlap all 21 over the other, so how much differences is in that area? Definitely here and then to this extent. To this extent, I believe could be similar. Okay, because this is simply providing an arm. It's like imagine a crane. Okay, so you cannot have really long length or short length. At the same time, it should have some flexibility. So, yeah, any other questions? Okay. So, yeah. So now the connection between the codon table and amino acids. So there are 20 naturally occurring amino acids. These are predominantly. There are of course few unnatural amino acids even in our bodies. But these are the 20 most common amino acids coming in protein synthesis. Sometimes, of course, the modifications are actually related. Okay. So these amino acids. And these are alpha amino acids, okay? So as the name suggests, these are amino acids. So you have an amino group and a carboxyl group, okay? And so these are alpha amino acids. Means that you have an amine group and the carboxyl group attached to the same carbon. Okay, and so you have a functional group R. Okay, so which actually make these amino acids differ from each other. Okay, and these are again L5. Okay, means that then stereochemistry is fixed. Okay, for all these natural amino acids, enzyme 11. What do you call it? L, So this R actually makes these amino acids rich in terms of their structure and function. So the table shows nicely they come in different varieties. Okay, the R is called side chain. So what are the properties? Small, large, charged, uncharged. Okay, charged, neutral. Let me call it. Polar, non-polar. Okay. Okay, and you need to think of other properties. Okay, so you can really group them in different varieties. Okay, so glycine is the simplest amino acids where actually the side chain R is under this proton. So you have just basically two hydrogens, and also the stereochemistry is absent because both are H. Okay. The next complex amino acid is alanine, where the R is CH3. Okay, and so like I said, these amino acids can be grouped into different properties: bulky or aromatic. Okay, and ionizable side chains. Okay, so alanine is the next one, and then valine, hydrophobic, okay, are non-polar. Leucine, even slightly larger chain than valine, extra CH, CH2, and then methionine having sulfur, and another amino acid which has sulfur is cysteine. Okay, that's polar side chain, and methionine is non-polar. Isoleucine and leucine differs, they have exactly the same molecular weight, but however the same chain connectivity is different. In one case it's basically the side chain is propyl, and in one case it's basically ethyl and it's two small groups. And when it comes to polar, there are serine, CH2OH, and threonine, both have OH groups. Okay, in them, and cysteine, SH, and then proline. This is a cyclic amino acids where actually this side chain is connected back to the backbone. Okay, and asparagine and glutamine, you have amide bonds in side chains, CO and H2.
Why is proline polar?
Proline polar. Yeah, sometimes it's difficult to say that why they are really put. Okay, so looks like maybe there is, so depending on the CH, all these three CH groups might not be really neutral because you have nitrogen and carbon. So maybe it's not three are not identical. Maybe there's a kind of dipole moment, I guess, small dipole maybe.
Should not be very low in quantity.
Yeah, but sometimes it's difficult to say that why they do. And aromatics, phenylalanine, tyrosine, and tryptophan all have phenylalanine and tyrosine have benzene ring. Tyrosine has even additional OH group, and tryptophan has bulkier aromatic group. Okay, so these three amino acids, phenylalanine, tyrosine, and tryptophan, are important for optical excitation. So you can actually use absorbance as one of the properties to recognize detect these amino acids, okay, as in polypeptides of proteins. So other group is positively charged residues, lysine, arginine, and histidine. So lysine has a NH3, okay, which can be protonated very easily. Arginine, again, positive charge, usually like group N, positively charged. And histidine, cyclic nitrogen containing ring, where actually the NH proton is very sensitive to pH, and very often when you have pH sense regions, okay, pH sensing regions in proteins, very likely that histidine is actually involved in the and another group is negatively charged R groups, that's aspartic acid and glutamate. So there is a carboxylic acid group, so which typically give protons at the physiological pH, that's pH 7.5. So they act as negative charges. So the side chains come in different varieties. You can have polar, non-polar, charged, uncharged, aromatic, reactive. And so for every side chain, so one of the properties which you do is that ionizability, whether acid base equilibrium we can undergo if there is an ionizable group. So side chains, if you see the table on the right side, on the last column, so these groups, cysteine, tyrosine, aspartic acid, glutamic acid, lysine, adenosine, histidine. So they have ionizable protons. So this is basically the pKa is given, so that you can really see that depending on what's the physiological pH, the pH around that amino acid in protein environment will really dictate that amino acid is what's its side chain state going to be. Okay. Of course, couple of interesting things. So side chains, for example, serine, threonine, and tyrosine. So they have OH groups. So these OH groups can undergo further what are called post-translation. Maybe I'll continue this in the next class. So I'll talk about a little bit more about more properties about these side chains, how they might be useful in certain proteins, and then grossly post-translation modifications. So this is the first layer of complexity, having 20 different groups. How the next layer of complexity will actually enrich further, okay, after the proteins have been made, okay, to make them either functional or make it non-functional. That's all for today, okay. I'm lazy in making assignments, but I need to make a couple of assignments, so hopefully you should get assignments in a couple of days. Okay. Okay, thank you. Any questions?
Sir, I have a question regarding the last class. In the last class, you have told us about the G tetraplex and the DNA triplex complexes. So in that complexes, like DNA and double-stranded DNA and RNA has specific works to do in our body. So that G tetraplex or the DNA triplex have any certain properties that they maintain in our biological system, or they are not normally made in the system due to the energy factor.
You're asking if DNA tetraplexes are other triplex structures.
No. I am asking that are they doing any biological role, or are they playing something important to some sort of gene expression or something biological information? Are they expressing, or they are forming inside our body due to some base pairing or something?
Yeah, one of the functions is that they provide rigidity, stability to the chromatin. Okay, for example, if you see DNA tetraplexes, okay, so they provide really rigidity. Imagine, let's say if you don't have, one is of course packing is very important. Second is the stability to the chromosomes, okay, and I think these are the important factors. Okay, also maybe to some extent, these are like capping layers. That's what I'm saying. So chromosomes, you have like an X shape, right? And then we always have small caps at the ends. So unless they are really stable there, okay, imagine they're actually packed together, okay, in nucleus, right? So there are many chromosomes really packed. So you need to really balance their structure, okay, and then accessibility of the genes which are required, okay. So this is nuclear dynamics. You know, it's a really big field by itself, that how these chromosomes are actually organized. You know, if you take any cell, more or less actually we know how these chromosomes are actually packed together in almost all cells. Okay, very tiny changes really occur so that certain genes are actually accessible for their expression. So it looks like these structures are already optimized their geometry in terms of the sequence on the length, so that they provide that structural rigidity. Okay, so that these chromosomes can be placed with respect to each other in a certain spatio-temporal fashion. Okay, which is required for the cells. So we can actually label each chromosome with certain color using fluorescent probes. And then what people have really looked at is that more or less the overall organization is actually fixed every cell. The moment actually it's disrupted, actually cells go awry, any major changes. But very small changes only really occur to really access the genes which need to be accessed in a spatio-temporal manner or in cell-specific manner. So in that sense, I would assume that at the ends of the chromosome really provide tight caps, okay, not only provide structural rigidity, but that will be helpful for placement of the interchromosomal distance. Okay.
Okay, sir.
The question in the chat. Can the videos? Okay, yeah. So yeah, I think Aditya is recording, so I promise that I'll post these lectures, videos online.
I have a question.
Only 1 and 2, so 3, 4, 5 I need to. And also slides I need to share. Yeah. Okay.
Can I ask a question?
Yeah.
So, sir, I am asking that for phenylalanine, let's say any protein, all the codons are same. For phenylalanine, there are 2 codons. So, if we use 2 different codons and the same phenylalanine came, so overall for the 2 different codons, the protein will be the working function of the protein will be different.
No. So that's what the slide I showed, that for a given sequence you can have different nucleotide sequences. So this is called redundancy, code redundancy. So let's say phenylalanine is coded by 2 tRNAs or 2 codons. Okay, it doesn't matter which codon is there in the mRNA sequence. So both codons will give you amino acids, phenylalanine. So in the end, protein, once it comes to the protein, you can't see that which codon it's coded. So both phenylalanines are exactly the same, but the codons are different. So mRNA different sequence, but these 2 mRNA sequences will give you exactly the same protein. This codon redundancy.
Okay.
Okay. And also depending on, so there are analysis, so certain codons actually appear much more frequently than other sequences. So you have actually corresponding phenylalanine, you have, let's say, 2 tRNAs. It doesn't mean that you have equal proportions of these 2 tRNAs. Imagine, let's say, if the codon frequency is also different, then you would imagine the tRNA frequency also will be different. Okay, so because there are codons with the frequency of the codons actually varies, and there are slight differences. Prokaryotes will have certain frequency than eukaryotes.
And are there any
Sir,
reason? Sir, are there any reason for the protein synthesis occurring along the 5' to 3' direction, not the reverse?
I mean, well, we name it as it occurs.
What? Okay. So are we leave or not? What? No.
Sorry, I got laptop got discharged. I start again. Hello, Aditya, can you hear me? So someone was asking question.
Yes, sir. I was saying that we can differentiate the mRNA from tRNA by their structural features. So how can we differentiate with the rRNA with these two, sir? rRNA, ribosomal RNA, sir.
No, distinguish means what? You don't have to really distinguish. RNA assembles along with other protein factors into ribosome as a macromolecular machine. Okay, so tRNAs are actually floating molecules. So they are synthesized. So then, whenever they have ribosome bound mRNA, so then each one will come and then check, okay, is it the right sequence or not.
So I'm thinking that mRNA is different from tRNA because of they have different structure. So is it correct?
Of course, like RNA structure in general, it doesn't matter what RNA is. Sequences, the structures are complicated. But tRNA, you have to distinguish is that tRNA comes with an amino acid covalently attached. Okay, I think at three parts.
Okay, yeah.
Okay, there is an amino acid specifically attached at this. And the size of tRNAs is again fixed. Okay, almost all tRNAs gross shape and size is fixed. mRNAs come with different variety depending on protein is small or large. You have different sizes. Ribosomal RNA again, I would assume that they are fixed. Okay, because you have to make just ribosomes. Okay, I think these are fairly large. Yeah, I don't remember the sequence size. But they bind proteins and then they assemble into only ribosome. Again, you have fixed sizes and shapes for ribosomal RNA. Again, they don't really exist independently. Means the moment they are made, they are actually assembled into ribosomes and they are released into cytosol. Okay, I think someone was asking like why the proteins are synthesized. I mean, mRNA and ribosome uses 5' to 3'.
Yes.
I believe once they're evolved, they have to really fix the direction. Okay, so actually it doesn't matter which direction they fix. Looks like early they fixed 5' to 3', and then they begin from 5', and obviously you need to have the end on the other side. Okay. Obviously, if they have somehow evolved beginning from, let's say, 3' to 5', maybe we would have named, then we would have asked like why it's not 5' to 3'. Okay, I think that's my simple answer. They have to put the direction on the fixed. You cannot have both directions. Okay. Imagine what would happen if ribosome makes proteins both directions. What are the potential problems?
Lot more proteins, the whole thing gets messed up.
Yes, because you have sequences, complementary to mRNA, but not in terms of protein. Second thing is that problem with stop codon. So if you read the sequence in reverse, okay, so I go to codon table. What you encounter is a big problem. Somehow they have to define stop, right? So if you imagine that stop, okay, so that's a nice corollary. Okay, can someone tell me looking at the table? So let's say there are 3 stop codons, right? If you read from 5' to 3'. Imagine ribosome is actually making 3' to 5' as well, okay? But the stop codons are exactly the same. So which amino acids codons in forward will act as stop codons for reverse?
Arginine.
So can you repeat the question? I cannot hear properly.
Imagine ribosome is working both forward and backward to make proteins. We know exactly how it works from the table when it works forward, right? But imagine it is synthesizing proteins backward as well and using the same sequence as stop codon. Okay? So which amino acids in the current table stop codons for the reverse direction?
Serine is one. Serine is there.
Okay, serine, what is the sequence?
AGU,
AGU. Okay.
UGA. So that would be like coming, if you're coming from 3', it will be coming like stop codon.
Exactly. Okay. So then you see that, serine is the coding template. Now, very often you will have how many codons are there? 2 codons. Okay. So that will be a stop, right? So then, unpredictably, you will have sequences which could be shorter or longer. It's impossible to predict. First of all, the length of the sequence and the size of the sequence, and you won't have any. Predictable sequence and the structures. So looks like somehow it needs to really stick, and then say that I need to have some rigid stop codons which can be there. Okay, so if it works in reverse, then it will be a problem. And second thing is that it will not. It will not, because so these are asymmetric machines. Okay, these are not symmetric. You can really think of this using sequences both ways. Okay, if you have a symmetric structures, then it doesn't matter. Sliding from left to right or right to left doesn't make change. But these are asymmetric machines. Okay, so these are, you can call what are called ratchets. Okay, so they go only one direction, because the mRNA strand, you know what is the backbone? Sugar, phosphate, and sugar linkages. So they are also actually important in pushing only one. Okay, so they won't slide back. Okay, so if ribosome has to work equally efficiently backward, then you would expect one of the properties that the mRNA sequence should slide both ways with equal efficiency. If it is equal efficiency, then forward sequence also will not be made, because then the amino acid is added, but it comes back mRNA, and you might add the same amino acid. Okay, so it doesn't really work. So somehow, because of the asymmetry, so it really ensures ribosome that the mRNA actually slides along unidirectionally, so that you have amino acids are actually added, and the polypeptide chain also actually moves unidirectionally in the exit channel of ribosome. Okay.
Sir, I have another small question.
Yeah.
So, sir, I'm asking that. So, in mRNA, who came first, ribosome or tRNA, during protein synthesis? So, when mRNA, ribosome came, then tRNA came, and the protein synthesis started, or other tRNA came and then ribosome came?
No. So typically, what happens is that this 50S and 30S to ribosomes actually I showed you. I think if I remember correctly, they can actually exist separately. This 50 and 30, because that assembly happens separately. Okay, so mRNA actually binds first 50S, then 30S comes on caps. So then tRNA comes, first tRNA comes, that methionine, okay, attaches, then ATP hydrolysis motions happen, sliding happens, then this second. So the sequence is that mRNA binds 50S first, then 30S comes on the other side, for example, and then methionine tRNA detection happens, and then further progress.
Okay, so sir, so when many ribosomes bind with the 50S mRNA bind, so tRNA came with that 5' direction and goes. So maybe in the middle somehow one tRNA came and bind the 3 codon.
No.
That's not possible.
Again, no. It's important that where does 50S binds? 50S binds at the beginning. Okay, so at the 5', you have even. That's what I told you. I didn't really go into details. So there are actually Tata box and things like that. Okay, there are important elements. Okay, even at the transcription level. Okay, and these elements are actually retired. They don't really code for protein right away, but they are actually adapter sequence. So your 50S actually comes and binds at the 5'. Imagine it cannot bind anywhere in between. Then you will have truncated protein sequences. Protein is always synthesized from the first amino acid to the last, okay? Typically, but always binding occurs 50, and then 50S binds 5'. Imagine then 30S comes, binds, your tRNA comes, sliding happens. So then this ribosome moves, let's say, along the mRNA. Then now you have 5' is free for the second ribosome to assemble. So ribosomes always slide. Okay? And occasionally, of course, they come off because of some reason. Then you have small peptide fragments. Okay? Or these are called truncated proteins. But typically, okay? So there is a sequence, there is a position where ribosomes actually begin to assemble on an You have a question.
So this is just like a follow-up on the 5' sequence question.
Yes.
So let's say I just switch the stereochemistry of all the chiral molecules involved. Does it still do 5' sequence or does the second
No, what you are saying is complementarity of exactly imagine you put a mirror.
Yeah.
Take everything. So they need to maintain the stereochemistry, everything in that mirror image.
Okay.
That's possible. Then mirror image board.
Yeah.
Hello, sir.
No, but then you have to really redefine in the mirror image board. Okay, you see, we call 5' because in the sugar, the attachment is 5-membered CH2OH.
Yeah.
Remember? Yeah, that's our labeling. That's still 5' only. But the stereochemistry is mirror image.
Yeah.
Okay. I'll show some examples of proteins, not the synthesis, where the structures are identical. I would say, let me put it like this. Enzymatic efficiency is identical for the mirror image enzymes and mirror image substrates. Okay? The world is exactly that.
Hello, sir.
At this level, you need to have every component mirror image. But it's possible. Yeah.
Hello, sir. Why nature choose this 3-letter code? Only the diversity, or there is any chemistry to get 3-letter code to amino acid?
No. Yes, Atish, good question. The chemistry is really central to biology. I mean, finally, the detection, okay, identification, binding, all these important factors actually bring up biology. And we say biology, we don't really talk about molecular terms, but everything really at the core, okay, chemical molecules. Obviously, chemistry is central. Okay. What is the second question? Chemistry is central. What are you asking?
The only diversity is there is any that 3-letter code to make amino acid part, like amine part making by this, acid part making by this, and R part making by this. There is that kind of thing is there or not?
I don't quite follow your question, but let me try to address. See, you have a genetic fingerprint. Means that
yes.
There's certain information here. I want to really make a copy of this because I like this. That's
yes.
what is actually reproduction and duplicity, division, and whatnot, right? But then you need to have that blueprint. That information is in DNA. Okay.
Yes.
So, but of course, to make this copy and to do all the day-to-day work, we need machines. So DNA itself is not going to make. So yes. Then those machines are nothing but protein molecules. But then how exactly they are encoded from this blueprint is basically this information translation from information translation from DNA to proteins. Okay. So in that sense, you call redundancy because somehow, yes, we are able to work with just 20 different functional groups to make complex machines. So we are using triplet codons. Okay. So one is not enough because then you will have only 4 amino acids.
Yes.
2 are also not enough, not rich enough. If we use only 2, so then how many? 4 into 4, 16 varieties of duplex, couplets. Okay. So then you need to reserve at least one for stop, just to say that.
Yes.
Then you have only 50.
50.
So it looks like somehow that's not enough. Looks like. Okay, so it made that little bit more variety. So you're using 3, but then 3 means 64 varieties, but we 20 are enough. So that's why wasting other codon. So you have to imagine, let's say you cannot have any triplet neither coding for an amino acid are using as a stop codon. Because then, okay, so if it is not coded for any amino acid, it should act as a stop codon. Because otherwise ribosome will stall, right? There is no tRNA coming. tRNA doesn't represent any amino acids. So then protein synthesis stops. So obviously it looks like to fill in those gaps, okay? So some amino acids are given rich variety, okay, special treatment. So leucine 6 codons, amazing, okay? And the tryptophan 1 codon and some other amino acids are 1 codon. Why? We don't know, okay? So that also shows that tryptophan is not a very, high frequent amino acid, okay? So you don't want to waste your tRNAs for tryptophan. Okay, what is the other amino acid which has only 1 codon? Methionine. So looks like again maybe they want to reserve only 1 for starting. Okay, and so yeah, leucine looks like hydrophobic amino acids have slightly larger number. Alanine has 4, threonine has 4, proline has 4, serine has 4. So it's not exactly amino, I would say. Yes. Yeah, cysteine has 2. Tyrosine has 2. Phenylalanine has also 2. Yeah. So it looks like only tryptophan answer. One other answer. Okay.
Yeah.
Good. Okay, that's all for.
Sir, I have a question.
Yeah.
So, sir, how much likely we have to expect that same amino acids form a peptide bond in a protein cell?
Same amino acids
forming a peptide bond. That is when we are getting adjacent same amino acids.
No, that's possible. Yeah. So sometimes after you have the kind of sequences, it's not that unreal. It's not that. Yeah. So occasionally, yeah, you have 2 amino acids, same amino acids ascend each other. It's possible.
Okay.
Yeah, it's possible. Like, for example, you can have alanines, or maybe glycine alanine, so things like that are actually possible.
Okay.
Okay, sometime, yeah. Okay, thank you all, and Friday we have our next class.
Thank you, sir.
Okay.
Thank you, sir.
Thank you, sir.